Well, everyone, thanks for coming in. This session is the future of genes and cell therapies. My name is Charlie, and I have Nathan next to me. We're the co-moderators of these sessions. We have a lot to cover today, so maybe we'll get right into it.
Yeah, thanks everybody for coming. I'm Nathan Yozwiak of Mass General Brigham's Gene and Cell Therapy Institute, and we're going to cover a lot of ground across the gene and cell therapy space. Maybe to kick us off, I'll ask Roger first. What do we mean by gene and cell therapies? I think increasingly the domain of what is covered in this space is growing, which may be a good thing, but it can lead to some definitional confusion. So why don't you set the stage for us and tell us what we mean by gene and cell therapies?
That's great, thanks Nathan. In general terms, gene and cell therapy, and from a gene therapy perspective, we're talking about platforms that deliver specific types of nucleic acids. It could be DNA, it could be RNA, it could be editors, using vectors or other types of nanoparticles for delivering of these nucleic acids. And then their ultimate goal is to either augment, decrease expression, or in the case of editors, to basically correct specific mutations. In terms of cell therapy, we're going to hear a lot more about them today. Again, broadly, these are engineered cells that can be modified using the same types of nucleic acids in terms of gene therapy, and they could be augmenting expression, decreasing expression, or changing in terms of editing the existing DNA in the cells. So gene and cell therapy are basically very broadly taken as these different platforms.
Good. Peter, so I guess there are a lot of cell and gene therapy being approved over the past several years, right? I think there's maybe 18 approved therapies in the US across 25 indications or so. Just wondering from your standpoint, from a regulatory standpoint, what has worked and what has not worked?
No, thanks for that question. I think we've seen a lot of progress over the past years in cell and gene therapy. I think what has certainly worked, I think, has been kind of an approach to trying to get data rapidly with therapies that are well characterized, and those have gotten over the finish line reasonably fast. In other words, work done early to make sure that the product is working, is well characterized, well manufactured, has gotten things over the finish line more rapidly than places where things have had to be re-engineered because people tried to move a little too quickly early on in development and have had to deal with things like comparability, et cetera. I think the other one piece I would say here that has clearly been an issue in the world of AAV gene therapies is that manufacturing still is a real challenge to get right and to do in a cost-effective manner.
Thanks. Moving on to cell therapy specifically, turning to you, Marcela. So there's a lot of success in the heme-onc space in the last several years, but it's been more elusive, developing CAR T therapies to treat solid tumors. Give us a little bit of background. Why is that the case, and what are some of the new approaches that you see that are going to be potentially disruptive in this space going forward?
Thanks for the question, Nathan. I think solid tumor is fundamentally in some ways different than hematologic malignancies, and I'd like to boil it down really to three things. So one is the tumor target, two is the T cell, and three is the tumor microenvironment. So one of the challenges is identifying the right tumor targets, but then in addition to just which target you're going to go after, there's how does the tumor cell respond to a T cell? And we and others have shown, for example, that solid tumors get killed by different mechanisms than hematologic malignancies. And so how do we then engineer the T cell, which is the second part, in order to most optimally induce cell death in the solid tumor? I think one of the challenges in our space is that there are so many different innovations that it can be difficult to identify a single mechanism that is going to address all of the issues. The third thing, of course, is the tumor microenvironment. And one of the things that we recently tested is having the T cell secrete additional bioactive molecules into that tumor microenvironment. We named it a T cell engaging antibody molecule, or a TEAM, which is really a dual specific tandem scFv that engages the tumor microenvironment and goes after a second target. And we've shown some, I would say, preliminary clinical data of efficacy in glioblastoma with a system where you have a CAR T cell, but that also delivers and manufactures a second molecule at the tumor site. And I think that I'm very excited about that technology and the promise of that, but I think it's also not going to be enough for all tumors. I think that we're going to have to explore ways of further enabling the T cells to stick around longer, and to potentially have additional functions to optimally lyse the tumor targets for solid tumor.
So Sam, maybe to you on just gene therapies. So maybe just talk about high level, kind of what are the different technologies, that can be used in the gene therapy, where we are in the field right now, and where you kind of could see in terms of the challenges that you're seeing right now, and perhaps on where we'll go, in foreseeable futures.
Yeah, happy to answer that. Happy to be on this panel with colleagues I've worked with over the years. Cell and gene therapy is going through its own waves, like any new technology platform or frontier would. For those who would recall the 1980s when the antibodies came to the fore, you had this initial excitement, in the 83, 84 timeframe. 85, you got the first protein drug approved. And by 89, everyone said, antibodies are dead. It's all about small molecules. And companies like Genentech even had a hard time financing. But then you saw this emerging resurgence where you now have tens of medicines that are game-changing, and there's more and more shifting towards biologics versus small molecules. And you're going to see the same with cell and gene therapies. There have been fits and starts, if you will. We had the initial wave of AAV therapies, but some issues with that platform in the turn of the century, and then at the turn of the millennium, I should say. And then you've had tremendous improvements. But if I look at science right now, we're making progress every day. You get gene correction at the right locus. You're getting very sophisticated cell therapies that can do the job of many biologics in one therapy. You're starting to get delivery to different organs in the body, not just the liver where we started out, but now to hematopoietic system, to the brain is the next frontier. So my fundamental feeling is, yes, we've had a few therapies approved so far, and they all have their own stories, but you're going to see a wave of new therapies now emerge as the field gets more mature, and you're going to see tremendous success because they're very bespoke therapies, they're very tailored, and they fundamentally address the challenge of biology that we're trying to solve. So it makes the world more deterministic as opposed to stochastic, and that's the world we're moving towards.
Good. Thanks. I think building off of that and turning back to academia a little bit, Roger, Sam spoke a little bit about the development pipeline, contrasting gene cell therapies with biologics and others that come before. With this optimistic view of the future of gene cell therapies, where do academic medical centers fit into this development pipeline?
Yeah. So I think as Sam alluded to, these therapies are very targeted, and they're going after a specific population of patients. And if you look at gene and cell therapy, academic medical centers are really ideal for these types of therapies to develop, because on one hand, academic medical centers is where the investigators are in terms of early stage discovery for the target. That's where many of the development for novel vectors and novel directed systems to the organs are being developed. And really importantly, and that's something that you can't replicate other places, is the fact that the patients are at the academic medical centers and the key opinion leaders along with the caretakers are there. So because of these gene and cell therapies are so specific in terms of the patient population that you have to target, you really need that expertise in terms of your clinical trial design, in terms of choosing the right patients who are going to benefit from these disruptive types of therapies. So from an academic medical center, I think you have all the ingredients to really go from early stage all the way to first in human. And many academic centers, including our own at Mass General Brigham, have set up institutes like the GCTI, which is really helping with some of the roadblocks such as manufacturing and regulatory alignment to be able to get to that first in human. Because at the end of the day, academic medical centers serve the patients, and getting first in human trials to the patient is really what's going to benefit them.
So maybe if we continue kind of like the industry and perhaps the academia kind of partnership, maybe Sam, if you can talk about, CRISPR Therapeutics have, all these kind of partnerships throughout the years, what are the reasons behind that and what makes a good kind of alliance?
Yeah, I think, they often say when you have a new platform or new field, it takes a village. I think for cell and gene therapies, it's going to take innovative partnerships between academia and industry, between biotech and big pharma, and many other types of partnerships to actually make it a reality. A couple I'll talk about, which is the medicine we just got approved, Casgevy, which treats severe sickle cell disease and thalassemia. That came after years of work. There were population studies done by the NIH or other institutes that said, what if we can elevate fetal hemoglobin to overcome the deficiency or defectiveness of beta globin, right, which is the cause for sickle cell and thalassemia. And populations were found around the world, including Syria and Saudi and Greece. And all that work led to this new types of screens that different institutions did. One of them was Boston Children's, and on the advisory board for Children's. And professors there or faculty there decided that they're going to do this screen to find edits that can elevate fetal hemoglobin, but it's not deleterious otherwise, which is where this notion of BCL11A enhancer edit came about. Because people were trying to edit BCL11A, but the enhancer was a new discovery. But an academic medical institution can only go so far. They can do some trials, they can start, but to bring a medicine with global reach, you're going to need a company. And CRISPR did the license that technology. But then even us as a small biotech said, we can do this in the US perhaps, but doing it globally is going to be challenging. So we partnered with Vertex, which have been a great partner. And now Vertex is commercializing this drug, approved drug, Casgevy, around the world, not just in the US and Europe as some of the more cutting-edge therapies typically are. So we are bringing it around the world. So I think for partnerships, what's important is understanding why we're doing a partnership. I think just doing partnerships for partnership's sake, oftentimes you've seen things languish because there's no attention to it. But if you say there's a specific reason where there's a particular technology in academia, or there's a particular way of doing trials, and companies can actually systematize that and do it in a more industrialized way, that creates a powerful basis for a platform or a partnership and we've done several of those now, some successful, some not so successful. I'll leave the unsuccessful ones for the bar later, but I think in general, we've seen 1 + 1 = 3.
Thanks, and Peter, turning to you. So maybe pulling a couple of these threads together, Sam's talked about some of the platforms as well as the time it takes to develop a new gene cell therapy. Of course, eventually, the regulatory process becomes a critical part of this. How is the FDA thinking about some of the guidances in the context of the speed by which some of these can be approved, now that we're gaining so much more experience? There's a platform designation guidance, for example, maybe you can speak a little bit about, as well as the START pilot program.
Right, so there are two different aspects. One is, do we have the right regulatory paradigm for the products that we're working on? And I would say that we are increasingly realizing that although the small molecule paradigm and the protein-based paradigm work kind of well together, gene therapies, particularly as we move towards gene therapies that are ever more platformized, and I think CRISPR is a great example of a technology where you're going to have products that are very similar to one another, except for a guide, perhaps. Even their delivery mechanism will be similar. And we have to find ways to move between products, allow innovation on those products without declaring a new product every time someone changes a small tweak to that. So we are in the process of working through this. We have the platform technologies guidance that's out to have platforms designated for a given sponsor's product. So if you have a product approved, you can have that designated platform. We're also willing to apply that even to products, a group of products under IND. So that's not in guidance, but it's something we're willing to do. But I think really to take that to the next level is to figure out how do we put together a regulatory framework that allows us to really move and individualize products and think about this almost more, there's kind of a more tool-like nature of CRISPR. It's not a device, don't get me wrong, we're not regulating it as a device, so it's not going to be a general tool device, but we do need to figure out how we move it along, and I think you'll see us over the next year or two really making an effort to get ahead of this, because I think, as I look 5 to 10 years from now, I think directly administered CRISPR is going to potentially transform a lot what we do, not just in the current gene therapy space, but also in the cell therapy space. And then the second part of your question is, what can we do to accelerate the pace of progress? And one of the things we learned during Operation Warp Speed during the COVID-19 pandemic was that dialogue between sponsors, particularly smaller sponsors, academics and or companies that might have only one product funded by venture, they need to be able to interact with the agency frequently if they're going to make progress rapidly. The old paradigm of type ABCD meetings, for those of you who aren't familiar, it's a very formalized meeting structure which takes 30 to 75 days to have a meeting. By the time you finish clarifying what the agency actually meant from the feedback you got, it can take 90 to 120 days. You can imagine that spending a quarter of your capital for the year on a meeting isn't the greatest use of time. So Operation Warp Speed, we gave kind of this idea of constant contact where we turned around questions within days. The START pilot that we're running is an attempt to do that for a select number of products where we have the bandwidth right now. We don't have bandwidth to do this for everything, so we have enough bandwidth to do it for a couple products, and what we've done is we've taken at the Center for Biologics for pediatric rare genetic diseases which cause a lot of disability or death in the first 2 decades of life. And we're giving them that same back and forth with the agency so that if they have a question, can we do X in our manufacturing process, or is this clinical design okay? They just send us an email or pick up the telephone and we get back a response within days. Hopefully that will make a big difference here. If it works, and we are measuring this using some consultants keeping metrics, if it works, we will try to expand this because I think it could make a really big difference, and it would pay for itself. You can get and do a back of the envelope MPV calculation, even we can do that, and show that if we could shorten development times by 10 or 20%, it would easily pay for the 50 or 100 new reviewers that we would need to make this standard practice.
Can I add something to that?
Yeah.
I think something like that, Dr. Marks, will be tremendously helpful, because one of the things people don't realize is there's a huge time lag when you make a design decision on a product to manufacturing it and bringing it into the clinic. For instance, if there is a change in a mRNA sequence, is that a new product or not? Or can we put it into the existing IND? It takes 18 months with the CDMO to make that. If you have your own manufacturing facility, obviously you have a lot more flexibility because you can say, well, we're making the new mRNA, not the old one, and change over the schedules and everything else. But as you know, with CDMOs around the world, you're limited. You have to then tech transfer something that's new. It's a 24-month cycle sometimes. And so oftentimes we're like, oh, should we make this change or not? Because it's going to cost us a lot more. And you're leaving something on the table in terms of patient benefit. But a program like the one you're talking about, it could make a tremendous difference, not just from a cost perspective, but from a speed perspective, and I think will lead to better products.
I'm so excited to hear you talk about the regulatory framework structure, because when I think of what some of the power of gene and cell therapy is, is the ability to innovate. You can actually modify the drug or the investigational product in a seemingly infinite number of ways, even without changing its primary mechanism of action. And so, considering a different regulatory framework for that, I think will be really transformative for the field to really be able to capitalize on all of the innovation that can occur in this setting.
No, I appreciate that. And some of this is us over the next years figuring out the right guardrails, because ultimately this is an area where we just have to make sure that we get it right, right? This is a place where there's not a lot of tolerance with genetic medicines for things going wrong. And particularly in our current environment, where it just takes one X posting and things can go to a bad place very quickly. So we just need to get it. We need to get it right. But we will get it right, because I think we have to, if we want to remain leaders in innovating here across the globe.
So maybe focusing on the rare diseases a little bit. I think there were a lot of buzz kind of recently regarding the DMD approval. If Peter, if you can talk about how the agency kind of latest thinking on the rare disease, kind of the approval process, where they're thinking about some of the, the guardrail around how you think about the certain indications, and perhaps also just to talk about the rare pediatric disease voucher, that particular program, and kind of where's the future with that?
So maybe I'll take that last first. So with any luck, Congress obviously cares about enough people care about it that Congress cares about it, because it looks like, at least even in the short term continuing resolution, they've included an extension of the pediatric review voucher program, at least through December 20th, which is when the continuing resolution goes through. And we'll see if that actually, my guess is that gives them an opportunity to potentially extend it further. But at least it's a reprieve. It's a 3-month reprieve. And that is hopefully something that's expected to pass, or else I won't be traveling after October 1st. The issue of rare disease, it's really important that we can get rare diseases right, because in aggregate now, we understand that there are thousands upon thousands of rare diseases affecting probably somewhere around 30 million Americans. And although not each of them are as severe as the others, there are any number of them that are severely disabling, not just for the individual, the child or individual affected, but for their whole support network around them. And so I think if we can get the ability right to go through multiple rare diseases, we'll get the ability to dealing with personalized medicine on a more global scale. Because ultimately, when we start to think about CRISPR, if we're thinking about genetic medicines on a larger basis, we're thinking about addressing a lot of different nucleotide changes for more common diseases too. So we got to get it right. And I think we look at it as a risk-benefit calculation in the rare disease world now. I think for the Sarepta approval, it was risk-benefit and uncertainty. It was what we knew about the safety of the drug, what we knew about the effectiveness of the gene therapy, and what we didn't know, and also the severity of the disease, all taken in the context of what else was out there. And that kind of leads us to come to a decision-making process. And I think no one would argue that some of the gene therapies that we're approving now are the absolute best possible gene therapies that there will ever be. But if we don't make progress, we're not going to have a field. People aren't going to. I think about it this way: sometimes you have to think about what will happen to a field if we say no, we will shut down things completely, potentially for years. That's not the reason to do an approval, but on the other hand, we understand the impact of our regulatory decision-making for entire fields, and that has to be factored in the background.
Great. Marcela, turning to manufacturing issues for the cell therapy space. We know there's the idea of decentralized manufacturing is a hot topic. Peter alluded to the personalization of a lot of these therapies. Is that where you see this field headed? If not, what is needed technically to see this? And then also, kind of a co-question for Peter, what would be needed from a regulatory standpoint for point-of-care manufacturing in the cell therapy space?
Yeah, I think there's a huge amount of interest in point-of-care manufacturing. A lot of academic centers have built cell manufacturing facilities and have a lot of the expertise within the center to be able to do this. In some ways, it's not that different than handling blood products, stem cell transplant, and so on. And so there's been a lot of interest all along. And I think initially, when there was first commercialization of CAR T products a little over 10 years ago now, that was sort of the hope, and then it became a very centralized process. I think with that came, there are some obvious benefits to it. The big pharma companies and large biotechs, make drugs that's their business model, and so they're very good at that. But it's also not that user-friendly, either to the patients, to the hospitals, or even, frankly, to those companies, because the process itself is so expensive and difficult to train and maintain. And so I think, as we've seen some of these larger companies sort of either scale back on their cell therapy manufacturing, it's seeming now that perhaps we should rethink about point-of-care manufacturing. It's also something that's been happening around the world, right? So in India, in Spain, in Brazil, and other countries, many centers are doing point-of-care manufacturing or sort of distributed manufacturing, where there may be one center within a country or within a region that does the manufacturing and then distributes. So I think that those models maybe reduce the logistical and operational burden, perhaps increase the safety in terms of just making sure that the right patient is getting their right cells, decrease the turnaround time, and I don't know if I said it already, just be more cost effective in general. So I think that there's a lot of interest in doing that. I think within the current US healthcare system, it's harder for other reasons, which has to do with reimbursement, liability. If there's not an FDA approval for a drug, then it's hard to get insurance reimbursement. And so even though it could potentially cost a lot less, if there's no one who will pay for it, it becomes sort of a non-starter. And so some of the issues, I think FDA has actually outlined some pathways for how this could happen from a regulatory framework, but it's still not a completely straightforward path to get an approval. And whether each center has to get their own approval, or whether there's some centralized, QA, QC that then oversees everything, which I think is a little bit more like what Galapagos is doing. I think there's a variety of ways to do it, and it's not yet clear what's going to be the most commercially effective while also being the most cost effective for patients.
Yeah, I think I basically agree with a lot of what you said. I think the issue, though, is that there are probably centers like Mass General, Penn, there are a number of them up around that are capable of doing this and doing it with high quality. The question is how many centers can do it with that kind of high quality, and what would sustain that. If I was going to think about a decentralized model, I would think, if we can develop devices that do this largely without a clean room, and that essentially have unit operations that can run, that may be the way you get this kind of in a decentralized model. But I do worry about this concept of quality control if we start to see this going to some of the centers that are not as familiar with the fastidious nature of the work. And this still isn't stuff that you can just like, oh, we're going to slap together a lab. It has taken places like Mass General, Dana-Farber, Memorial Sloan Kettering, Penn, taken them a while to really get their facilities to a state of high readiness to produce these therapies. And I think short of putting it in a box, which a number of companies are doing, that is a way to have distributed manufacturing. And maybe that limits your options, but maybe then you have tiers where you have the innovative centers, and then you have some that can produce it closer to the patient for less cost. And I worry about just the shotgun idea of cost being less at academic centers is just taking it, because what actually then is sometimes not figured in these are the costs the overall cost of having a trained staff that you have to keep there all the time, despite the fact you might be making only 10 or 20 products per year, which could end up, although it may seem economical at the beginning, it may turn sour after you realize that you're not making enough product to recoup the cost.
Can I add something
Yeah,
to
please.
that? I think it's going to be product by product. I almost think decentralization could be harmful to the space in a way because you need certain scale to achieve high quality. And because otherwise you're spending a lot of money just keeping a plant open. The place where it may make sense is autologous CAR T, right? Because you're innovating fast, but there still you need innovation on the device front.
Side.
I mean, some of these devices that you use for CAR T manufacturing, it's like an old Pontiac Bonneville. It's like the dials and everything else. You look at it, there hasn't been a lot of device. You're still using, the dials and the prodigies that are from the 80s. So I think on the autologous CAR T, if you get like this cassette concept, which happened in molecular diagnostics with Roche, right? You had the same machine, you just had different tests, you had a different cartridge and you put it in, and everything else was a closed system. Until you get to that point, it's going to be very difficult. But for mRNA-based therapies, lipid nanoparticles, you want to do these at scale. I mean, allogeneic therapies, we make our allogeneic CAR Ts here in Framingham, not too far from here, and we can produce thousands of patient doses out of 10 runs in 2 months. And with that comes a lot of practice, and you're turning the crank. It's a standard system, and I think you get to better quality, better standardization, better predictability. And so I think this is a debate we have all the time in the industry about decentralization versus centralization. I think for the industry to mature, you're going to need these central hubs that define quality parameters in manufacturing for the industry.
I want to follow up on that, right? I mean, you talk about allogeneic and then obviously autologous, but there's also kind of the different other cell types, right? NK cells and different processes like iPSC. Just thinking about like, maybe just describe how high level, how are those newer approaches or perhaps different from the ones that we're currently kind of used to. And two, perhaps a question for Peter from the regulatory front. Like, how is FDA kind of thinking about that?
Well, yeah, we're working with different cell types. There's T cells, alpha beta T cells, gamma delta T cells, NK cells, and then there's a source, healthy donor derived or iPS derived. I think still some uncertainties about which one's going to be the winning cell type, if you will, but people are reverting back to T cells now because they seem to be the most cytotoxic against cancers, especially in the oncology space, and mainly towards alpha beta T cells. What's emerging though is that you can make these with iPS cells, but there's the heterogeneity of healthy donor derived T cells actually benefits the function in many ways because you're trying to kill these cancers. You don't know there are different levels of maturity and differentiation. So we are getting to standardization. We are getting to understand which ones are the best. But what's going to really move the needle, I think, are these edits in these T cells. We're finding new edits in T cells, or we're having T cells secrete various factors, as Marcela was talking about before. An example is IL-18, which Carl June described, and there is other edits like Regnase-1, which we're practicing, and they make these cell therapies much more potent, 10x more potent than the first generation. And that's going to be the step change versus the source of cells or the type of cell, in my opinion, in really making a move against these diseases.
I mean, I just would add that seeing the field of cell therapy next to the field of genetically modified cell therapy, what you see is the problem with unmodified cell therapies is that unmodified cells do what the heck they want. And the beauty of making a genetic modification is you force them, that becomes the dominant characteristic of these products, and so it makes a huge difference. It's why it's such a struggle in the unmodified cells. You have to have potency assays. The cells tend to, even for a given cell type, they tend to change over time. There are so many struggles, and I have incredible empathy and admiration for those who have tried to move those forward, because it's challenging and you want to bang your head against the wall sometimes. Whereas for the genetically modified cells, they're forced into doing things, and just as Marcela is trying to make multiple edits, that can really this can make a huge difference, and you can have predictability in a way you just don't have from genetically modified cells.
Plus just tracking them, right? Like doing PKs, PDs. It's when you have something to follow quantitatively and assess function, I think it's really powerful.
Yeah, and looking ahead to new applications of cell therapies, maybe we can talk specifically about a couple types of indications. I think the autoimmune space is getting a lot of attention right now. Marcela, maybe you can talk a little bit about how what we've learned so far in cell therapy may lead us to some new interesting developments in the autoimmune space. And then Peter, maybe you can follow up with how the regulatory standpoint, it can leverage what's been learned already.
Yeah, I mean, I'm an immunologist by training, and really the holy grail for so long has been how do we control immunity and tolerance. And in some ways, like cancer and autoimmune disease have been thought to be two sides of the same sort of conceptual coin. And what I find so interesting is that CAR-19 and to some extent also CAR targeting BCMA, in both cases T cells, autologous T cells. Actually, I'm so autologous T cell centric that it didn't even occur to me to think about other cells until you mentioned it. But just that going after the B cell compartment seems to be effective for both lymphoma and for autoimmune disease. And I think it's really interesting because there are potential ways of doing this with Tregs, but even just getting cytotoxic alpha beta T cells, autologous T cells targeting the B cell compartment seems to really reset the immune system and potentially antigen presentation in patients with a wide variety of autoimmune diseases. I think there's still a little bit of a question around a couple of things, like for example, the trials that we know of with autoimmune disease still include lymphodepleting chemotherapy ahead of time and at doses that are not typically used in these autoimmune diseases. So I think there's potentially some impact of that conditioning chemotherapy just in the beginning. And the fact that for reasons I don't understand, the toxicity profile seems to be improved in autoimmune disease. Now, maybe it's a matter of antigen burden, that patients with lymphoma or leukemia have a higher number of cells expressing the antigen, whereas in autoimmune disease, you have normal numbers, but they're just spewing out more antibodies or having more of a physiologic effect mediating the autoimmune disease. But in any case, the toxicity profile seems improved. The duration of B cell aplasia seems to be shorter. And so there's this concept of this immune reset that seems to be happening by a combination of the chemotherapy and the CAR T cells. So I'm really excited to see what's going to happen there. I'm hoping that there are just as a scientist, I'm hoping that there are some basket studies, because I think if it have to be individual studies for each different rare autoimmune disease, it's going to be very difficult. And I think already there's enough evidence to consider either small trials or single patient use for patients with very rare autoimmune diseases. I'm thinking like, odd paraneoplastic syndromes, just going beyond sort of the ones that have the greatest market. There are so many patients with really rare autoimmune diseases that could potentially benefit. So I'm excited to see where it goes in this way.
Yeah, I think it's a very exciting area, and I think that there was a little bit of a temper, the enthusiasm when we saw some of these secondary T cell lymphomas. I think that is calming down significantly because A, they seem to be quite rare, and B, many of them are not related to the therapy, but are just happening. And it turns out there's a low incidence, but a real incidence of T cell lymphomas in people who have B cell disorders that are not related to the CAR. So there are probably going to be some rare CAR-related lymphomas, but they're pretty rare. And I think in this autoimmune space, if you look, many of the diseases that we're dealing with they may not be cancers, but they have outcomes that are nearly as bad as cancers with the existing therapies. And I think whether it be anything from severe rheumatoid arthritis to systemic sclerosis, it may really be a a boon if we can move these forward. So hopefully we'll see those move ahead.
Maybe just to follow on that, but can you talk about perhaps the benefit kind of risk you're expecting in the autoimmune disease space? How is that kind of different from the cancer kind of indications?
I think it goes to understanding the disease that this is being used in, and if for instance, lupus is a disease that it can be mild, it can just cause a skin rash. On the other hand, I've watched people die of lupus because they can develop inflammation of the lining of the brain and the brain itself, or it can essentially destroy the kidneys. Those are really bad outcomes, and to prevent that, I think we would consider that I don't see that very significantly different. Preventing that is not that different than preventing a lymphoma, dealing with a refractory lymphoma, in that you potentially have a life-saving outcome. So there's probably going to be, there will probably be a difference, because at a certain point there would be, do you want to use a CAR T cell to help prevent the butterfly rash in lupus? No, you probably wouldn't want to, because that's like not a high enough benefit risk. On the other hand, if somebody is starting to develop failing kidneys and neurologic symptoms, I think we would think about that very similarly as an oncology indication, and there's probably something in the middle. And I think, again, something Marcela already mentioned is the fact that we don't see some of the part of the problem is when you give CAR T cells to people with lymphoma, some of these have some pretty significant side effects. You need an ICU around, whereas here, it looks like possibly some of this could be outpatient therapy pretty easily, because you don't have those same reactions.
Great.
So maybe we'll open it up to this question up to everyone. We'll start with Roger. No conversation around cell and gene therapy is complete without a dedicated question about costs, which is hanging over the whole field. What's your vision and optimistic ideas for the future, and how we can collectively improve the cost question?
Yeah, I think innovation is really going to be the key. We need vectors that are more targeted, so we have to use less of them by logs. We need cells the same way, that are more targeted, and then we need less of them, so the manufacturing cost has to go down. And I feel that's really where we're going to need to focus our attention, not only from a cost perspective, but more importantly from a patient safety perspective, to have much more targeted therapies, and that will reduce the cost and improve safety.
Marcela?
So I almost wear like two hats in this area. So as a translational scientist, I think about, in some ways, the valley of death has gotten deeper and longer, which is frustrating, because it used to be that the biggest step up, or the reason for the valley of death, was around the first vector manufacturing or the first cell manufacturing. And now it's really become more about how close is it to an approval, and how many patients have been treated, and what's the signal, and what's the market sort of potential value. And so I think that it has extended the runway. I think it has so much potential, though, because CAR T, at least, is not, the effects are usually not subtle. And so you don't necessarily need hundreds or multiple hundreds of patients in order to get to an approval. But the other side, and so I think that's overcomable, and I think we have to be patient and think about how much we're really changing the landscape every time we learn from a small trial. But then I also wear this other hat as the director of the cell therapy service with my colleague Matt Fregault, and there it's about giving commercial CAR T product. And that is so difficult, because the final products for standard of care are just so expensive. And so, handling the payers and the cost for that is really a challenge. And I think that there's going to have to be some coming into some resolution of that in the field. And I'm not exactly sure what it's going to be, whether it's going to be distributed manufacturing or changes in how the cells are manufactured themselves. But that is a real issue. We can't get to this point where we have these wonderful therapies, but nobody can actually get them. That's not why we're here.
I'll try to be quick. I think it's going to be a combination of moving towards directly administered products and figuring out a way to improve our manufacturing technologies so that there's automation used in manufacturing, potentially leveraging artificial intelligence and next generation manufacturing technologies.
Sam, final word?
Prediction, the cost is going to come down by 3x in the next 5 to 7 years as a combination of innovation, scale, and automation.
Great, thank you.
Thank you.